Very good. And also, I will shortly introduce Sonia and Thibaut, who will be our speakers for today. So Sonia was the former FORCIS postdoc, and she works in the fields of ecology and diversity of plankton. She works on the influence of climate change foraminifera distribution across the global ocean, also with questions of carbon cycling, and does comprehensive data synthesis and analysis, which you will shortly be hearing about. And our second speaker, who is Thibaut de Garry-Belteron, he works in Serege in France, in the CNRS, and he was one of the principal investigators of the FORCIS group, and he works in paleoceanography and micropaleontology. He also does the ecology of foraminifera, and he works on reconstructing the past environmental changes from foraminifera assemblages, on geochemistry, and on traits. And he also works on quaternary paleoclimates. And now I will let you get on with your presentation.
Thank you, Camille. Thank you, Nicolas, for your kind words. I'm very happy on behalf of all the group of Cesab, FORCIS, who was, we really thank you for your support along these years. And I think well, it was a long journey, but it was a successful one, as we hope to show you. And I will try not to get a too strong accent. So next slide, if it does work. So we will just show you. So the group was called Foraminifera Response to Climatic Stressors. I was co-PI with Ralph Schiebel, that I also thank, who has been very active in supporting the group. And it's a group effort. So yes, it's very important for that. So the outline of the talk. So first, I'm not sure you are all aware of what are foraminifera. So we'll have a very short introduction of what are forams, why are they important for climate studies, and I will introduce the main goals of the FORCIS project and the research questions. And Sonia will show you how we handle the FORCIS database with the help of Nicolas, who is there, and Xavier. We show you after some of the results we've obtained during the last years, and also some future direction as for example the Cesab return project that will submit soon. So what are foraminifera first? So I'm not sure you are aware of that, but when you swim in the open ocean, you can find maybe some in the plankton. There will be some of these bugs that you see at the top. They are typically 100 micron to 1 millimeter. They are quite rare in the seawater. It was part of the questions we wanted to address. They are protists, so unicellular, and they build this shell, which is in calcite and carbonate. And you can find foraminifera in the upper water column, something like between 500 meters deep to the surface. And their shell is very well fossilized. You can find sediments bearing foraminifera almost everywhere in the open ocean. So you can see on the left a picture of a sieved sediment that comes from the tropical Atlantic, where you have mostly these bugs. So each individual is composed of different chambers. And to the right, you have the living example of a foraminifera picture taken by Julie with these spines and some symbionts. So historically, the foraminifera have been very well studied in the fossil world because you can find it in almost any sediment core. And there has been since the end of the 19th century a lot of studies. and it's been used for reconstructing paleoclimates. And what I show in the bottom left is a distribution of the surface sediments, where there have been some countings of the different species of foraminifera. So this is a compilation that has been done by the group in Bremen. And you see that almost everywhere we had some data. This is what this data of the diversity of assemblage of foraminifera is used to reconstruct past paleoclimates. But on the other hand, in the modern ocean, there was almost no comprehensive analysis of the ecology of forams. You can see, for example, this old study by Alan Bay, where you have for this species the map of the distribution, where it was something like 10,000 forams per 1,000 of cubic meters, and here 1 to 1,000. So we didn't have really a grip of how many forams there are there in the open ocean. And it was one of the main impetus to start this work, trying to get this to tackle the ecology and the diversity of forams. So why you should stay and not disconnect right now? First, because Sonia will will talk after, but also because foraminifera are exceptional in the sediment record. They have they are continuous archive of speciation in the marine record, and we can find them in marine sediments. They are quite important in the biogeochemical cycle, particularly the biological pump. They are something like about 10% of the modern carbonate flux to the seafloor right now. It's a matter of discussion, and it's also a lot of some works that we're going to pursue in the future. But I said that already. There are excellent paleoclimatic archives. So we use the assemblage, the diversity, but also the geochemistry of the shells. And last, there are some sentinel species of climatic changes, and that's going to be the main focus of the topics. And what is particularly interesting for the foraminifera is that they live in the water column, and we have some records of the water structure of the ocean using these bugs. So one historical study that it was a part of the discussion at the beginning of the project was this old paper more than 50 years ago by Bill Rudiman, who was comparing the marine sediments distribution of forams. So this is all the dots here are modern sediment surface sediments, and these two triangles are plankton nets. And in the 70s, he already assumed that he compared the composition of the assemblage of foraminifera in the plankton net to the closest analogs of in the sediments. And he showed that there was a shift of the plankton net compared to the closest analogs. So even there, he was assuming that it was an evidence of a modern warming of the North Atlantic Ocean more than 50 years ago. So this is pretty much what we've been trying to do, but at the global scale. And it's something that was also studied since the last glacial maximum 20,000 years ago. So on the left, you have a panel with the latitude. So this is the North Atlantic from the equator to 70° north. Over the 21 kilo years ago is the last glacial maximum, and you have different assemblage of foraminifera. And what you see is that they do change across the deglaciation quite rapidly in the tropics. And still, we are missing. So in the sediment to the modern, but we are still missing the modern component. This is pretty much what we are trying to do. And some studies that have been doing the latitudinal diversity gradient, but Sonia is gonna tell you more about that. A bit of history of the project. So it wasn't very successful at the beginning. So we were rejected twice for the FRB-Cesab call. I would like to thank Alison Specht, who was the previous Cesab director, who has been supportive. And we also we had some what really was really the trigger is some discussions with the group that evolved, and also we got some seed funding from the INSULEF, and eventually in 2018 we were funded, and it was quite a bit about that. First meeting in June 2019. Sonia was hired in 2020, and she got a 2-year position after with the Max Planck Institute to work on the project. And I would like to thank the 3 guys at the bottom left, Maud and Nicola, swear, for their help. It was a really long, fun work. And I will let the grant to Sonia for all the remaining of the session. Sonia, do you want to share?
Yes, thank you, Thibaut. I'll share my screen. So as Thibaut said that, I mean, this project is having as a goal, especially to assess if the abundance of these species is decreasing or increasing in the modern ocean, if there is any change in the habitat or any vertical movement of species toward higher latitudes, or if they are keeping the same habitat as before, or if there are any species that they present different sensitivities to the global changes, and also to assess the present and the future climate response of these species using different methods. So to do that, I think Thibaut and Ralph, they contacted different people having different expertise, and they ended up with this cool list of people that they contributed efficiently to bring this project to the success. And also I would like to thank Nicolas Cajasius, that he contributed also a lot during all these years with the database construction, the R code analysis, statistical analysis. And thank you all for that. Then, so together with the group, we tried during the project to evaluate the biodiversity changing of the calcifying zooplankton in response to multiple stressors by building an ambitious database, gathering planktonic foraminifera information from the water column from historical till today. And we did this to improve our understanding about the spatial and the vertical distribution of these species, and also to explore the effect of the main stressors such as temperature and ocean acidification on the planktonic foraminifera distribution and calcification process, with a focus on the present and near future ocean impact. So to understand these changes, we use a surface sediment database, FORCIS mainly, to compare with the modern data that we have in FORCIS. And then we use also some future simulation to see the impact of the environmental changes on the ecological niche of the planktonic foraminifera. So I will detail everything later on in the slides. So to do so, first of all, data regarding the foraminifera diversity and distribution from the global ocean were collected. They were sourced from published and unpublished data, and sometimes provided directly by the data contributor from FORCIS working group or through personal networking. So around 130 references reporting historical and modern data coming from 4 type of sampling devices, which are the plankton tow, the continuous plankton recorder, that is presented here, sediment traps, and also water filter that were derived. Most of the historical data set were digitized from published tables or plots, while the modern data were sourced mostly from contributors' data file and imported directly to the database. up to now, we have a huge database that we kept it open, and we are actually welcoming any contributors to submit data until now. So the database is huge, covering both spatial and temporal magnitudes, and it contains around 1,300,000 counts coming from 160,000 CPR samples, 9,000 sediment trough samples, 22,000 plankton net samples, and around 300 PAMP samples data. So here you have a map showing the position of the whole data set that we have within our database. And also the FORCIS database covered data compilation from 1910 to today, and the different magnitudes are well covered within the last 30 years. And for certain regions, especially in the North Atlantic Ocean, we have a temporal coverage that reached 100 years. So as any big data compilation, handling it sometimes could be tough. And thus, with the Cesab data scientist, Nicolas, again, we worked on the development of an R package to help downloading, filtering, unifying the count units, and also making basic plots like this one here now to help the database users to visualize the data. So the package is online and ready to be used just in case you want to play with it. So our database, in our database, we have samples that they were collected with different mesh size. Like here, an example, you have like in the same position, two people that they are sampling with different nests that they have different mesh size. Like the first one is having 100 micrometer of mesh size, and the second one is having 200 micrometer for mesh size. If the two person having the samples, they want to compare their samples, it won't be it won't be possible because the first one will catch much more forms than the second one. So this will impede the direct comparison. And for that, we generated a technique to normalize the absolute abundances for the plankton net, CPR, and PAMP data to 100 micrometer mesh size. And this technique was developed using samples fractionated into different size fractions, and from the relationship between the size fraction and the cumulative abundances that is having a mechanism and shape, we derive equations that allowed us to normalize any abundance to any size we prefer. Then after standardizing the number, the abundances to 100 micrometer of mesh size, we analyze the data to answer our research questions and especially to assess the response of planktonic foraminifera to anthropogenic changes. So in order to better understand the total changes of abundances through time, the abundances of the major species of planktonic foraminifera in the North Atlantic and the Arctic Ocean is calculated for each decade and per latitude. Like the first panel is presenting the species occurring between 0 and 30 degrees north, and the second one is species present in between 30 and 50 degrees north, and in the last one are the species occurring in the very high latitude between 50 and 90 degrees north. And at the species level, the results show that the abundances of all the species, most of the species of planktonic foraminifera are decreasing gradually through time, especially for subtropical species in the mid-latitude, such as Critrata and Eutertrae and Testaculifer. In addition to that, the mid-latitude from 30 to 50 degrees north is becoming more diverse with the presence of tropical and subtropical species. At the global level, this abundance is showing a significant general decline that goes up to 24% of decline, especially at the mid-latitude. As we observe this decline in planktonic foraminifera abundance, it is naturally leads us to investigate how these species richness changes within this ecosystem. So to do that, the latitudinal diversity gradient derived from the FORCIS database is presenting high peaks in the mid latitudes and lower diversity in the low and the very high latitudes. When comparing the species diversity in FORCIS, presented by the dots in orange, yellow, and pink, to the FORCIS database on the surface seafloor sediments presented by the gray dots here and gray line. Using the same taxonomy level, the latitude and diversity grading patterns show some differences. So the diversity in FORCIS is getting higher from the mid to high latitude compared to the FORCIS database. This high species richness in the high latitudes encountered in FORCIS could be due to the species poleward migration to poleward migration. So this behavior was observed also for many marine plankton species, including coccoliths and theropods, that increasingly expanding its range into polar oceans. So which species has migrated and which species was not migrating? This is presented in the next slide. So analysis on the changes of the spatial distribution of planktonic foraminifera in the North Atlantic and the Arctic Ocean before and after 1990 show a northward migration of most of the species, such as G. inflata, G. rubescens, and the Tertrae and G. cultrata, with 10 degrees of displacement to the higher latitudes. The other tropical and subtropical species like G. titula and G. sacculifer and G. ruber are also presenting lower northward migration between 4 degrees and 10 degrees to the higher latitude. However, species like G. ruber and G. obliquloculata are moving southward, but also they were not showing any changes in the abundances, highlighting their insensitivity toward the climate change. These results push us to investigate more if their distribution through the water column was also changing. So to do that, the depth at the maximum abundance of species of planktonic foraminifera was calculated for periods before 1997 across the latitude bands. So here in the first panel, you have species occurring between 0 to 30 degrees north, and the violin plots, the first half in the violin plots is presenting the depth habitat of these species before 1997, and the second half is after 1997. And we did the same for the other latitude, covering for the species living between 30 and 50 degrees north. What we can see here, at least for the low latitude, that species like Callida and Crassiformis exhibit a significant deepening. The species here presented by the red dots, correlating with environmental changes such as rising in temperature, water column stratification, or any shift in food resources. These species are moving deeper, likely to align with deep chlorophyll maximum that is now becoming a bit deeper in the modern ocean. However, at the mid-latitude, the response is mixed. So you can see that some species, they were displaying a deepening, and the other ones, they are now shallowing. However, many young-bearing species appear to be more stable, constrained by their resilience on surface trophic conditions, and because they need light and they need to occur at the surface of the ocean and they cannot go deep. So the story is not finishing here. And to assess the future evolution of the environmental condition in the different latitudes in the North Atlantic Ocean, where the different planktonic foraminifera habitat changes were occurring, we use temperature and saturation state of seawater with respect to the calcite, and we did some future simulation to determine their variability in 2050 and 2100 according to the mid of the road scenario, as predicted by the Earth system model. So you have here for the actual niche are presented by the gray dots, all the foraminifera occurring in FORCIS, and in a purple color are the future occurrence of foraminifera in the 2050, and in pink one are their occurrence in 2100. The first panel presenting species occurring in the low latitude, and the second panel is a species occurrence in the mid latitude. So our results show that in the North Atlantic low latitude in 2050 and under the middle of the road scenario, most locations where georuber, for example, and testicular fur have observed will fall outside the current planktonic foraminifera environmental niche due to the warming under this condition, which no modern planktonic foraminifera species are observed. And in that case, population decline would be expected and are not be replaced in the low latitude, unfortunately. In the mid latitude and also in the high latitude, the panel I'm not presenting it in this slide, the species occupy colder and less acidified conditions, as you can see here for the purple and the pink dots. So these species, warming and acidification 2050 and 2100 will not bring these species to be occupying niches outside the current plankton foraminifera environment in the modern state, and thus the mid to the high latitude existing planktonic foraminifera may be replaced by other planktonic foraminifera species that adapted to warmer and more acidified environment, as it is already observed through the poleward migration. In addition to that, some species will be occurring in omega calcite that is predicted to decrease to values where the foraminifera could not precipitate their shells. We think that the different abiotic and biotic mechanisms can be invoked for this decrease in abundance and ecological niche changes, which are the reduction in diversity in low latitude in waters where temperature exceeds the upper limit of this thermal range of some species, the discordance in the phenology of planktonic foraminifera with their prey for the carnivorous species, also the migration of species toward higher latitude or deeper depth, and finally the effect of acidification and dissolution on these species. So our study shows for the first time that planktonic foraminifera are able to respond quickly to the environmental changes, most through migration and abundance decline, even though the high latitude ocean are warming and acidification is occurring faster than elsewhere. It is the low latitude ocean that plankton foraminifera as a whole will suffer the most, because population decline will have difficulties to be compensated by suitable plankton foraminifera immigrants. So these changes will have also different impacts, especially for the paleo community, using the forams as a proxy for paleo studies, and thus an update of some knowledge must be applied, such as the transfer function. Also, the poleward migration to areas where omega calcite will decline will induce the reduction in calcification for some species, and this will induce the appearance of probably naked forams, and the loss of the shells, which constitute a powerful archive for geochemical and paleo studies, for example. And all this will impact the carbon cycle, as about half of the calcite produced in the modern open ocean is through the originate from the planktonic foraminifera. This work has also provided an opportunity to update our understanding of the current distribution of planktonic foraminifera in the global ocean. Pioneering studies such as the one of Bracho and Beetal led to the foundation of, by creating the first comprehensive maps and defining key biogeographical provinces like tropical, subtropical, temperate, also subpolar, polar, and upwelling region. And building on this legacy, the FORCIS database reveals significant latitudinal, vertical, and abundance change of planktonic foraminifera. And to understand how these shifts manifest, especially, we have plotted their current distribution across the global ocean, highlighting where these species, presented by the blue dots, are occupying which latitude. So all the color bands are the provinces generated by Beital, and through this work, we are trying to see if the species are occupying the same provinces that were presented by Beital or not. And since we saw a lot of changes in diversity and abundance, this work will update what the previous studies were stating. This is not all. We also assess the vertical distribution of planktonic foraminifera across the different ocean basins. Here, I present only 2 examples, focusing on their depth habitat in the North and the South Atlantic Ocean. As you can see here, all the species in the Minor Ocean are occupying the upper 150-meter depth of the water column. So one of the questions that we were also interested to answer during this project is, are these changes will continue happening in the future or not? And this work has been done in collaboration with Annabelle Go, which is a PhD student at the Boston University. She used niche models such as generalized regression neural network and the environmental string models, calibrated with the FORCIS data and modeled environmental data to provide an independent estimate of planktonic foraminifera distribution and abundance in the future. So these 2 models were applied to predict the future abundance change for 6 species that I'm presenting here, and for 3 different climatic scenarios. So here you have the different outcomes for the 6 species coming from 2 different models. So the dark colors are the ASM model, and the lighter colors are the ERNN model. And as you can see, wherever the scenario is, the abundance will continue dropping in the future. This is a bit scary, but also if you look at the diversity pattern using the same models and using and assessed on the 41 species that we have in FORCIS, the global diversity will continue also dropping, especially in the low latitude, where we will have the more pronounced diversity decline over, the whole latitude gradient. On the light of these diversity and distribution changes in the modern ocean, and given that these species play a key role in carbon sequestration, we were particularly interested in assessing the contribution of these species in the carbon and the carbonate standing stocks. And using the data of FORCIS, we converted the planktonic foraminifera abundances into carbonate and carbon standing stocks. And here are 2 primary result maps illustrating their spatial distribution across the global ocean. For the carbonate standing stocks, the mean value is approximately between 0.5 milligram per carbonate per cubic meter. And for the organic carbon standing stock, the average is around 0.1 milligram of carbon per cubic meter. Overall, the planktonic foraminifera contribute to an estimate between 1 to 10% of the carbon standing stocks among carbonate producers in the modern ocean. But which species dominate the inorganic carbon stock at the surface? And here we present a map illustrating this. So the standing stock are primarily driven by few key species, such as T. sacculifer in the low latitude, Biloides, and Pachyderma in the high latitude. However, some of these species, particularly sacculifer, is showing a concerning decline in abundance in the modern ocean. And this decline poses significant challenge as it represents the loss of species crucial to carbon sequestration in the ocean, potentially impacting the ocean role as a carbon sink and its ability to mitigate climate change. So to wrap up, this project has delivered the first comprehensive database on the distribution and the abundance of planktonic foraminifera across the global ocean. Also, the FORCIS database has enabled to analyze large-scale patterns and trends, shedding the light on how plankton foraminifera respond to environmental changes. So we observed a decline in abundance, diversity shifts, and evidence of vertical migration in the water column for some species. Also, our projections explore how these species might adapt under future climate scenarios with a focus on their ecological role, particularly in the carbon cycle. So these results are the effort of the group members. Here are some pictures I wanted to share with you to see how we were working at the Cesab in Montpellier all together to do this work. So everyone contributed and everyone worked super hard for this project. But when it comes to fun, we had a truly blast. We explored great restaurants. We shared plenty of love. We enjoyed many memorable moments together. FORCIS also has an inspired project where several, has inspiring a new project and collaboration and opening door for future research. Here I've listed some of this initiative. And at the end, I would like to thank all the FORCIS members for their help, and as well the FRB and Cesab for their support. Also a special thank to Maud, Nicolas, and Nicolas. And many thanks to the Serge and the Max Planck for supporting me through these years. Thank you, Camille and Nicolas, for the invitation and for giving us the opportunity to present our work. And finally, thank you all for your attention.
Wow, thank you very much, Sonia and Thibaut, for this presentation. Maybe you can stop sharing your screen so that we all see each other now. And yeah, that was super interesting. And so now we will have some time for questions. So I invite everybody, all the participants, to please raise their hands so that your little icons can line up and we can define, and I can distribute the speech to whoever wants to talk. And we'll have a session, yeah, we'll have like 20 minutes, up to 20 minutes to have a discussion. And if you're not able to talk, you're also very welcome to write your questions in the chat and I will read them out loud. Okay, Bastien, hi.
Hi. Hi everyone. So thank you very much, Sonia and Thibaut, for your very interesting presentations. So I'm wondering, you talk about the possible consequences on consumers, and so maybe possible consequences of shifts. Did you think already to investigate more concretely this possible impact on the trophic network? And if yes, how do you plan to manage this?
Do you want to go, Thibaut, or?
No. I think we are, we might disagree, the two of us, but yes. Sonia, you want to have a first stab?
Yeah, actually, worms are sentinel species that they are not really, eaten by like pelagic species, but they are actually giving us the information about what's happening in the ecosystem, as they are co-living in the same environment as other important species for the food web. And so if the forms are shifting or declining, so the other species should do the same. And already a lot of studies, they were showing, like for copepos or for some coccoliths, that they are showing the same trend and they are migrating to higher latitudes. Of course, we can also dig into the details and like try to analyze other databases on other calcifiers and try to compare to our database, but there are already some work that is published. Yeah, I don't know if I answered your question.
Yeah. So
okay, and you mean already at this scale also, at this global scale, there is already some studies that investigate the consequences of shift of foraminifers on the rest of the networks?
I think one point is that they are not that important in the trophic in the trophic structure. They are more they are quite diluted in the open ocean. I don't some, I'm not sure. I think maybe some foraminiferists might disagree, but they are really important in terms of inorganic carbon and for the ballasting, for example, of the export of organic matter and inorganic carbon to the seafloor. But in terms of organic, the trophic web, I'm not sure they are that key. And that's when you look at the numbers, the abundance, and etc. It's not spectacular.
Okay. I was just wondering because you mentioned that it might be an effect on consumers. So, okay, many thanks.
But Sonia might disagree. Huh? But
No, I agree that they are not eating by big like fish and so on. They are finding them in some booths, like they estimate it accidentally, but they are not sure that they are eaten by these species. But as I told you, they are sentinel species, and they are informing us about the other species eaten by like fish, if they are declining or if they are perishing. Yeah, so they are good indicators, let's say.
Yeah, thank you. Many thanks.
Does anyone else have a question? Nicola has a question.
Nicola.
Yeah, well, I won't get into the discussion on if they are or not into the food web. I mean, I guess, you know, they exist and they have probably quite a biomass. So somehow they are part of the food web, and even if it's indirect. But I'd like to ask you about the evolution. Like, I mean, I don't know about the generation time for those foraminifera, but it must be week or weeks. And so I would expect them to evolve and adapt in the Darwinian sense to some environmental change. And they are probably among the best candidate for what we call evolutionary rescue. Like, and so when the condition will become, you know, toxic or not that good, they might evolve. And so can you tell something about that or your guess?
Who start?
Yeah, go ahead.
Yeah, well, in terms of yeah, so you're right. Typically, the life cycle, one forum lives for about 1 month to a few months. So it's quite it's quite short as a lifespan. So the first thing, when you look at the paleontological record, foraminifera species didn't evolve very drastically in terms of shape over the last million of years. For example, rubber, that one species that is quite abundant, has been there since at least more than 10 million years. So there's not a lot of plasticity in terms of the shape. So it doesn't mean that in terms of the function, it didn't evolve over time. So this is and with regard to what you call evolutionary rescue, I'm not sure we have the so up to now we couldn't. One way would be to do culture. For example, it's been done, for example, for the coccoliths, I know, and looking at the acclimation of the coccoliths and of the coccolith of course, sorry. And for foraminifera, it's tricky because the foraminifera up to now were not. You couldn't culture them. You were just picking them into the seawater, and they were just growing, and then there was no reproduction until Julie arrived. Julie Meillan was part of the group of the meeting, and now she can grow. She can grow foran for at least 2 or 3 generations. So, and in terms of molecular biology, I don't think we have any element on this evolution at very short time scale. So I'm not sure it's a very good question answer.
No, I mean that's great. I mean, I thought that you would be able to actually cultivate them and look at how fast, you know, they evolve in the lab. And even if they don't change in shape, you know, they might change in terms of how they actually can handle the temperature or whatever. But I mean, that's clearly, I mean, given that you suggest that there is a critical role in terms of carbon, you know, storage for the following years, it's probably one of the most important questions is they won't disperse, they won't, you know, follow the climate as you claim, but they might have an evolutionary response. But just let it there for now. Thank you.
Yeah, but if, for example, for the coccolithophore, there are some resistant species to acidification. It's been shown, for example, in the South American margin. There are some strains that are resistant, but I think for the forums, we don't know yet. And Sonia,
we know a little bit.
to...
I mean, yeah. I mean,
yeah,
the change is nowadays is very fast and happening quickly and continuing actually changing, and they are trying to track these changes. And I'm not sure if they are adapting at 100% or not. Some species they were showing, I mean, a huge sensitivity, other less, and two only that I saw that they were not migrating or dropping in abundance, and probably they are more adapted because they are living actually in a very hot, warm environment. But yeah, as Thibaut said, and now I'm not aware of studies on the genetic for the modern species to see if they are adapting well or not, or if there is any emergence of new species.
Okay, thank you. Does anybody else have a question? Otherwise, I have one, but it's less ecology and more data science. So maybe if somebody else has an ecology question, maybe they should ask it first. Okay, well, I'm going to ask my question and we'll see. I have a question based on the database that you guys put together. And I don't know, it was just a general question about how your experience of doing that. Was it complicated? What does it take a lot of effort to sustain? Like, since you said that you're going to keep it open, and would you, I don't know, do you have any tips or recommendations? Does it foster a lot of collaborations? Do people use it, or is it principally for you? Like all of these questions, if you have anything to say about it, I'm happy to hear it.
Yeah, thank you, Camille. Can I answer the question? Okay. So, I mean, I started the postdoc with that, gathering the information, put them all together. And I think Thibaut started just, I mean, before me, a few years before me, trying to collect the first data. And when I arrived, I tried to check this data and also add more. Well, to be honest, of course, as any start, it's a bit difficult, but with the group, it was okay because it was not only me who is collecting the data, the group is also providing data. A lot of people, they have in the group, they have a lot of data, which is cool. When I have questions, it's easy to answer, but also they have a lot of connection, and through their personal connection, we got more and more data. Gathering was not that difficult, but harmonizing and structuring was the most painful part of the work, especially when you have a database like FORCIS. You have different sampling devices, you have different mesh size, a lot of taxonomical biases, and luckily we had a lot of experts in FORCIS, like Gertjan Bruma, I think he's here in the chat, that helped a lot with Ralph Schibel for the taxonomy. Xavier Giraud, he helped a lot with Nicolas for the structuring of the database. And of course, everyone contributed together and collect the data and do the quality control. And as people are still publishing data, and it's always interesting to collect this data and put it together and put it to FORCIS and to keep growing it always. And now with the R package, I think it will facilitate a lot the handling of it, or even if it will grow a lot. And hope that it will grow a lot. And for the collaboration, yes, we receive some people that they want to use the database, and they are interested to use it. And yeah, it's now used also. Which is cool. Yeah.
Awesome. That's good to hear. Thanks a lot. And so Michael has a question.
Yes. In the beginning, you said that somebody who would use a different mesh sized net would get different results. So could it be, for example, that there is also a shift in the size of foraminifer? I know this from different assemblages that the size in the fossil record, they differ substantially. And how do you cope with this problem that maybe the amount of specimens is the same, but they just have a shift in size, which you didn't catch if you normalize the kind of data?
Yeah, thank you for your question. Very interesting. Actually, most of the species, they are adult at this, a size between 100 and 125 micrometer. And this is the minimum mesh size that we use to catch forams, although some historical data, they use lower values of mesh size. It's true that some species, they are becoming smaller or bigger due to environmental conditions, but they won't drop to 100 micrometer less than that, and I think it will be small, but even the loss will not impact significantly that. Yeah, I don't know if Thibaut wants to add more about that.
Well, just that you did some work to do some standardization. There was, I think, a couple of slides in your talk. There was a paper was trying to tackle this shift in the mesh size from larger size in the early in the 50s, in the 60s, where they typically had this mesh size of 300 microns to 500 microns, and now to smaller sizes. So what Sonia did and what we did is quite the best that we could do, I think, trying to correct for that. But you're right that in some way you can't if some forums go below there is no way right now to correct that using this kind of extrapolation. I guess if you use maybe some machine learning techniques, it might be possible if you have at least one or two a set of samples for benchmarking your machine learning, but it's a tricky question, and we don't have we did the best we could, and I'm not sure there is other ways to correct for that. And I'm open to any suggestion.
All right. Any last questions? Well, I don't think so. So I guess that's it. Thank you very much for your presentation. It was super interesting, and we were happy to have you. And thanks to all the participants for being here. I hope you had an interesting session. I'm going to stop the recording, and yeah, if you're going to hang out so that we can debrief a little bit, that'd be great. So yeah, as I was saying before the presentation, everybody stay posted for the next Cesabinar, which will be on January 23rd with the School Reef group about the variability of coral reefs. Thanks and goodbye.
Thank you. Bye-bye.